of the newest member of the omics family. And so first we need to know something about what a metabolome is. And basically a metabolome is everything that's not a nucleic acid, polymer, or a protein. So a lot of different things. And usually the goal of metabolomics is usually to study how these metabolites, the relative amounts of these metabolites are changing in response to various conditions like a drug treatment or a genetic mutation or environmental conditions or something like that. And so what makes metabolomes interesting is that, unlike genomics, transcriptomics, and proteomics, which phenotypes aren't always associated with the things you're seeing, metabolomes in their very essence are basically a snapshot of the chemical reactions that are going on in a cell. So it's a very descriptive and intimate portrait of cell physiology, basically. And so I kind of think of this as sort of a little, increasing in what I like to think of as complexity going down this way. It's like a genome sort of sets the boundaries on what's possible for a cell, and then through transcribing certain genes and alternate splicing, you translate, you make proteins, you can post-translation modify these proteins to give them activities, but at some point you generate molecules that catalyze chemical reactions. So the chemical reactions that are being catalyzed are all of these metabolites. And so kind of as you move down this way, you're getting a more and more accurate picture of what the cell's physiological state is. But along with this, you're also getting much more chemically complex. So it's more difficult to study the whole picture when you're looking at metabolomes. So there's a variety of things that metabolomes can show you. I'm going to go through some of these in detail. We can skip over this. And then some common approaches to analyzing metabolomes. So first, I have to mention that mass spectrometry and NMR are two indispensable tools for doing this. That's just because of their ability to provide very detailed structural information on a lot of different types of molecules. We basically can't do metabolomics without these guys. So usually these analytical techniques are coupled to some sort of separation, and that's just because metabolomes are very complex chemically. So usually you have to fractionate in some way and analyze a certain sub-portion of the metabolome you're interested in. And there's two general types of metabolomics, untargeted and targeted, and we'll get into what those are in a minute. I'll give specific examples. But there's some major challenges to metabolites, metabolomics that I've sort of hinted on. And it's basically variability in the physiochemical properties of metabolites. Makes it difficult to develop one approach to analyze everything. And it's challenging to adapt to the single cell level, but we're getting there, and I'll show some examples of that. And still another big issue that we need to deal with is understanding more about the spatial distribution of metabolites. I'll also talk about some recent technologies where people have been employing imaging mass spectrometry, various forms of imaging mass spectrometry, to get at this last question. So I'm going to talk about a few examples. And so this paper about 10 years ago was from Ben Cravat at Scripps, who's sort of one of the, helped develop the field of metabolomics. And so this is a very interesting study. So basically what he did was he took a wild-type mouse and a mouse that had a double knockout of this specific enzyme. And so what you can do is you grow these mice up, you kill them, you take the brains, crunch them up, and then you compare the analyte to metabolite levels in the two different samples. And so what this is showing is basically the mass of the compounds on one axis, the elution of these compounds off of the column on this axis, and then the z-axis is showing the ratio of these metabolites in the double knockout mouse to the wild-type mouse. So anywhere you see a big peak is basically a metabolite that's accumulating to a very significant extent only in the double knockout mouse. The implication being that since you knock out both copies of this functional enzyme, all of these peaks are substrates of this enzyme. So this experiment effectively shows you the endogenous substrates of the enzyme you're interested in. So this is a very difficult question to get at. And so using this approach, they saw a variety of metabolites that were known substrates of this enzyme, but they also identified several novel substrates. And so they proposed that these were perhaps neurotransmitters that weren't known at the time. And so using a very similar approach, this more recent paper used basically this untargeted metabolomic strategy to identify a cancer biomarker. So when I say untargeted, basically what I mean is they're trying to look at everything and identify gross differences. So this experiment is basically identical to the one I just showed you, except in this case there was some genomic information signal intensities for the metabolomes of those two conditions. Basically, all of the 1,000 species they detect, only 3 of them had higher levels in the mutant. And they're basically all different forms of this metabolite. It's basically a side product, or a product that's made by this enzyme when you mutate it. And so to show that it's really a cancer biomarker, they did this metabolomic analysis on wild type and mutant cells. And they showed that all these different primary metabolites had very similar levels, but this particular metabolite they identified on the last slide always seemed to be elevated in these cell lines that had this mutation. So stable isotope resolved metabolomics is another approach, but in this case what you're actually doing is following the flux of atoms through metabolic pathways. And so doing this, you can kind of study how different pathways interact with each other. And so it's very difficult to study it's very difficult to quantify the steady-state concentrations or changes in steady-state concentrations of metabolites because there's generally not much change. But if you can take a sample and spike it with heavy atom isotopes, both of our common analytical approaches are actually sensitive enough to detect when these isotopic molecules are moving through the pathway. So basically what you can do in this case is, for instance, you have this, it doesn't matter what the compound is, but you can distinguish the light one from the isotopically enriched one. And so you feed the organism or whatever a bolus of this enriched compound at a certain time, and then over time you can follow how these isotopically enriched molecules are moving through the pathways. So basically it gives you kinetics on how these heavy atoms are moving through pathways. So you can get a lot of very detailed information on this. We'll skip this slide, so I want to get to the single cell example. So this was actually done by Sweetler at UIUC, developed this method where you couple capillary electrophoresis to electrosporanization. And so basically you can do this at the single cell level. You can take a single cell, chop it open, and then detect up to hundreds of metabolites in a single cell. The technique's very sensitive, it's quantifiable, you can scale it up. Here's showing the chromatogram basically of 2 different single cells. They look very similar, so they're detecting similar intermediates. They can actually take the cell, chop it up into portions, show that levels of specific metabolites are different in different regions of the cell. They can do MS-MS, further structurally characterize. So I'm going to try to get through this real quick. Imaging mass spectrometry this review here covers a lot of different new techniques to do this, but basically the idea is to get a spatial picture of how metabolites are distributed in your organism. It's not quite at the omics scale yet. There's a lot of spatial resolution issues. This guy at UCSD, Peter Dorsey, been doing a lot of this. And this picture basically shows 2 bacterial colonies. This guy right here is about to eat this one, and so this one is making this molecule called surfactant, which is an antibiotic. And this interaction is triggering the cell that's about to eat the other one to make a couple of different proteins that are involved in eating it. So that's pretty cool. And then, so finally, last technique I want to talk about is a very new technique called NIMS, nanostructure initiator mass spectrometry. What's cool about this one is that you can basically deposit this matrix, and you include these initiator molecules, which basically aid in the ionization of the analytes you're interested in. And so what's cool about it is that you can put your sample directly on top of this surface with these initiators. And then what's cool is that it's a soft ionization technique, so you can detect proteins and larger molecular weight things. You can, by doing this in a slightly different way, you can get pretty good spatial resolution. Right, it's very minimal sample handling. All right, be able to get to the important stuff. Okay, good. So in this recent paper, so using NIMS, they were actually able to, so this is showing a mass chromatogram of a single cancer cell. So these are all different compounds that they can detect in a single cancer cell. And they're basically just comparing this NIMS technique to other techniques where hundreds of cells were pulled together. So you can see there's actually excellent metabolite coverage. Another thing that's cool about SIMS, so this panel is showing they're analyzing 1 microliter samples, I'll be done in a minute. They're analyzing 1 microliter samples of 2 different biofluids. This red is blood. The blue and the green samples are the same urine sample. And you can see that once again, from 1 microliter, you just spot on top of this on this surface, you can detect many different metabolites. What's cool about this is these 2 urine samples. This is the same urine sample, but all they changed between these 2 conditions was the identity of the initiator. And so you can see that different metabolites appear. So there's hopes that by modulating the initiator, you can get better coverage of the total metabolome of the state. It's very nice, very sensitive, and you don't really need much material. We'll skip over this and we'll get to the conclusions. So I hope I didn't talk too fast. I've mentioned that metabolomes provide rich information on the physiological state of cells. Right now, solution-based methods are currently the more advanced of the techniques. Other people are starting to adapt these metabolomic principles to single-cell level into imaging mass spectrometry techniques that actually allow us to see the spatial distribution of metabolites. So.
Yeah, can you roughly explain how we get image from mass spectrometry?
Oh, okay, yeah, so I kind of skipped through that slide. Well, basically what you do is you have a sample, so in the case of MALDI, you have your cell or your tissue, you can cryosection tissue, you can do a variety of different things, but basically you have it like on a two-dimensional surface, and so for NIMS, you would just place the section on top of the nanostructure. For MALDI, you would have to actually place a matrix on top of your sample, but then you just raster scan, you scan like a laser across. And so in each position, you blast it with a laser, record you get a mass spectrum. At each position, it's just basically like pixelating it. So each position you get a spectrum, and you go through the whole thing, you generate a spectrum in each pixel, and then you reconstruct an image, of where the metabolites are on, your two-dimensional space.
So, so a single image can only tell you a single small molecule distribution. It can tell you about more than one molecule.
It can tell you everything you can see in the mass spectrum. So, if you're lucky, if you use the right matrix, if your sample behaves, you can see hundreds of things. And so doing MALDI is difficult because it requires that your analyte crystallizes a certain way with the matrix. And so there's all kinds of artifacts that can give you false quantitation signals because the analytes crystallize in a different way. That's what's nice about NIMS. There's no matrix, very minimal sample handling. And it looks from at least some of the data they've shown that you can alter the identity of that initiator molecule as you go into detail if you want to get different metabolite coverage. So you use one compound, maybe you see 50 metabolites. You want to see other metabolites, you can maybe change that compound and see different things. And there's no matrix artifacts in that case. So it's pretty nice.
Okay, so all the omics techniques which are more distance to be techniques you can actually use for clinical studies because you're using blood samples and
Yeah.
samples which are going to be is it like to use it because
Yeah. It is. Yeah, I didn't have time to go into the examples I showed where, identifying substrates for an enzyme. Another thing you can do is, one of the examples I didn't go through is they actually had two cancer cells. They treated one with the drug, and they didn't treat the other one with the drug. And basically from doing some relative quantitation between the two samples, they were able to identify that in the sample that was treated with the drug, they were actually inhibiting fatty acid biosynthesis. So you can actually identify the effects of drugs or toxic compounds or things on, the distribution of metabolites that you see. And from what we know of primary metabolism, we can go back and say, well, maybe, that's why these guys are sick. We're altering the flux of metabolites. We're stealing metabolites from one place and putting them somewhere else. We're altering the flux of compounds or atoms to a different place, and that's a problem. The thing is, with the powerful thing is you can actually globally, the idea is the hope is to globally look at this, to see how multiple pathways interact with each other.
might be more important for like infectious disease or like rather than like
Yeah, I think it's just to show you where anytime there's a difference that you think is important. If there's a phenotypic difference, there's going to be some difference in the level of metabolites in the two. There has to be. Because that's really the descriptor of what the cell is at that moment in time. So yeah, it depends on what you can do with the information once you get it. I'm sure it's probably pretty easy to use some of these techniques to find differences. But just because you see a molecule is higher in one condition and not the other, it doesn't really mean you understand why it's that way or what that really tells you about how you can fix the problem. But I think the point is the methods are sensitive enough and thorough enough that there's a lot that you can do with it.
okay. So thank you so much.